Genetics is concerned with the transmission, expression, and evolution of genes, the molecules that control the function, development, and ultimate appearance of individuals. In this part of our lecture, we will look at the rules of transmission that govern genes and affect their passage from one generation to the next. Gregor Mendel discovered these rules of inheritance in 1866. However, only in the year 1900, three botanists, Karl Correns of Germany, Erich von Tuschmuck of Austria, and Hugo Duvaris of Holland, finally defined the rules that govern this transmission of traits from parent to offspring. But some historical controversy still exists as to whether these botanists actually rediscovered Mendel rules by their own research or whether their research led them to Mendel's original paper. Here is the list of certain more discoveries from the year 1903, where Walter Certain hypothesized that the behavior of chromosomes during meiosis explained Mendel rules of inheritance, thus leading to the discovery that genes are located on chromosomes. In 1913, Alfred Sturtevant created the first genetic map using fruit fly. He showed that genes existed in a linear order on chromosome. In year 1927, Muller showed that genes can be mutated artificially by X-rays. In the year 1930 and 32, Fisher, Wright, and Haldane developed the algebraic foundations of our understanding of the process of evolution. In year 1943, Luria and Delberg demonstrated that bacteria have normal genetic system and thus could serve as models for studying genetic processes. In 1944, Avery, MacLeod, and colleagues showed conclusively that deoxyribonucleic acid was the genetic material in all cells. In the year 1953, James Watson and Francis Crick finally cracked the structure of DNA. In 1968 and 73, Arbor, Smith, and Nathans discovered and described the enzymes restriction endonucleases, which could manipulate DNA through recombinant DNA technology. Finally, in 1972, Paul Berg was the first individual to create a recombinant DNA molecule. So our journey had a long way. Genetics can be easily divided conventionally into three subclasses: classical genetics, molecular genetics, and evolutionary genetics. Classical genetics describes the chromosomal theory of inheritance, that is, acquired characteristics in an individual. Molecular genetics talks about the study of genetic material, that is, the structure of DNA, the expression of proteins, the regulation of proteins. However, evolutionary genetics discusses the mechanism of evolutionary chain, which include concepts such as variation, speciation, etc. Now, these three major area genetics largely covers. In classical genetics, we have the Mendel's principle, the cell division processes of meiosis, mitosis, sex determination in various organisms, sex linkage, chromosomal mapping, and cytogenetics, that is, the study of maps of genes. In molecular genetics, we study about what is the structure of DNA, what is the chemistry of DNA, the process of transcription, the process of translation, the mechanism involved in DNA cloning, the science of genomics, the control of gene expression, how DNA mutates, what are the repair mechanism, and what is extra chromosomal inheritance, that is, what are the other organelles in a cell which carry chromosomes. The third section is evolutionary genetics, in which we study about population genetics. the quantitative genetics as we call it, in which we study about Hardy-Wiener equilibrium, assumption of this equilibrium, evolution and speciation. Now genetics is an empirical science. We all know that science follows a scientific method wherein an observation leads a researcher to propose a hypothesis and then to make predictions from that hypothesis and to test these predictions by an experiment. The results of the experiment either support this hypothesis or refute the hypothesis. If the experiment refutes the hypothesis, a new hypothesis has to be developed. And in case the experiment support the hypothesis, the researcher designs further experiment to try to disprove it or make it into a theory. One important point here is that a very good experiment need to always have a control. Now we studied about Lamarckism, wherein we studied that there is a theory of acquired characteristics. The giraffe can have long necks as evolution said, that over the time they acquired long necks. However, evolution proved that only those giraffes could survive which had long necks because they could reach out the trees wherein they could find food. And this characteristic got incorporated into the giraffe population. But can we study giraffe and can we study the population of giraffe since giraffe takes a long time to give birth to a new giraffe? So what is the need for model organism? The need for model organism is an organism that can regenerate in a very small time and they can reproduce a very large number, and we can actually study the organism in various mutated forms. So we have here the adult female fruit fly Drosophila melanogaster, in which we can study mutations of eye color, bristle type and number, wing characteristics, which are easily visible when they occur. Then in bacterium, we have the famous Escherichia coli bacterium. Then can you name all these other organisms? We have zebrafish, we have rat, we have yeast, we have C. elegans, we have Arabidopsis thaliana, and the number goes on. Now why they are called as model organisms? Because we can easily study their characteristics in a small time limit. So classical genetics, Mendel did the same. His model organism was Pisum sativa, the garden pea plant. And the year 1866, he carefully controlled breeding experiments where he studied the alternative forms of Pisum sativa, and he worked on 7 traits of the plant, each with 2 discrete forms that affected the attributes of seed, pod, and stem. The seeds could either be round or wrinkled, the pods could be full or constricted, yellow or green, the stem could be tall or dwarf, and so on. So if we see the glossary or the terminology of genetics, we understand that genes are a unit of inheritance usually occurring at specific location or loci on a chromosome. And if there are alternative forms of a gene, these are called as alleles. If there is only one copy of a gene, you call that as haploid state. If there are 2 copies of gene, you call it as a diploid state. If that allele is dominant, that is, it masks the presence of a recessive allele in the phenotype, you call that as a dominant allele. And if the allele that is masked in the phenotype by the presence of a dominant allele, that allele is called as a recessive allele. So there were 2 major rules of Mendel's that were of segregation and independent assortment. Using Pisum sativum and the tall and dark character, abbreviated as capital T and small t, when the fertilization was done using the haploid gamete, in the diploid form we saw Even if there was a heterozygous condition, the diploid still had the tall trees. So Mendel crossed tall and dwarf pea plants, demonstrating the rule of segregation, that alleles segregate during fertilization. A diploid organism with two copies of the gene for tallness per cell form gametes that all have the dominant allele. Cross fertilization produces zygotes, and when these form the present zygote, the plant is still tall, indicating that the capital T is dominant to the recessive small t allele. So, how do we study these allelic forms? How was it confirmed? It was confirmed by studying the mutants that were produced as a result of these mating experiments. So, mutants finally led to the concept of chromosomal mapping. So, in Drosophila, research was done and mutants were studied, and finally, a chromosomal map was prepared in which you could specify the location of all the alleles using map units. So, centromere is the constriction in the chromosome, and the genes or the alleles that are present distant to each other are measured as map units. So here we stop and we will continue further on talking about classical genetics and the rest of the Mendelian rules in our next section.